Hello. This is a tutorial video on how to use the automatic splice site and exon definition analysis server found at splice.uwo.ca. It is a bioinformatic resource for evaluating mutations for its effect on splicing-related binding sites. I am ASSEDA, first author of the paper describing this resource, which is going to be published in Human Mutation in 2013. You do need to have to register this resource to use it. You should receive the confirmation email within 10 minutes to activate your account. Once activated, you enter. You'd have to put in your information, but I already did that. And then we read the terms of use. And then we go on to the main input page, and this is where we find a mutation based on gene name, which you enter here. But you can also submit your own sequence, go by mRNA accession number, or by RSID. For my example today, we're going to be using a BRCA1 mutation. Here we input an HGVS notation mutation. You can put in a genomic coordinate, which is, I find, the easiest way. This website, ASSEDA, is based on a previous version called ASSA with the previous splice.uwo.ca, which used to be HG15. Now the coordinates have been updated to HG19, as has all the databases for RSIDs and gene names and coordinates. You can also put in other HGVS notation variants, such as IVS notation. This should be the exact same mutation. You can also do CDOT. And they do not have to be point mutations. You can also put in deletions and insertions. The window range is how many nucleotides out the program will scan for other sites, natural or cryptic, surrounding the mutation entered. In this case, I'm going to look a little bit further, 200 out, as I know that there is an 87 nucleotide site that gets used due to this mutation. Here's the list of weight matrices or ribbles that we use. By default, acceptor and donors are automatically on, but there's also branch points, ribbles, different splicing enhancers and repressors, and mice, if that's what you're interested in. These new acceptor genome and donor genome ribbles are weight matrices that are based on over 100,000 different acceptor and donor sites. So I'm going to activate all 4 of those. And here, this is very important, it's the molecular phenotype prediction by exon definition. There's a new feature with ASSEDA, the exon definition, where it will try to predict which exons are most likely to be formed due to the mutation. And this needs to be selected for this to do that. Moving on, then we get a list of all the different RNAs for BRCA1. I know for a fact that this is important for the cDNA or the IVS notation, or because if you pick a different splice form, those might be off. So I know we're picking this one. You can click Submit right now, or you can go into Options, which gives you some flexibility. You can change the threshold to go below 0 or above 0 if you'd like. You can, for CDOT notations, you can start from the open reading frame, which is default. You can also start from the start of transcription. A new option that we're also using for the exon definition feature is to include potential enhancers and repressors in the calculations. For now, we just have SF2 ASF and SC35, and you have to decide if it's going to be an exonic enhancer or an intronic suppressor. The reason why this is off by default is because if there is no enhancer or repressor involved with this exon, then you end up skewing your results. So we submit. The mutations are analyzed dynamically, so there is a lag between submission and reporting of results, about 30 seconds per run. If you make a mistake with your inputs, say you put a T here and it's not a T in the reference, the website will tell you, and tell you basically to guess again. It'll have kind of a guess on what you meant, but it doesn't read mine, so just double check and make sure you get it right. And since it was a donor mutation, we'll look at the donor, and we see that the natural site, position 0, the natural site, has been abolished. And there's some nearby cryptic donor sites, one 2.3 bits, 87 nucleotides out. The strongest one is negative 190, so 196 nucleotides upstream. This is upstream of the acceptor, and the splice order is inconsistent with the mechanism of splicing, so it cannot be used. Here we have the phenotype prediction, and this is where the exon definition information is. Here we see the RI totals, which is kind of a measure of the strength of the total exon based on the donor acceptor in a gap surprisal. You see that the natural exon is being abolished, and the most likely one is this 87 nucleotide downstream donor and the natural acceptor, position 0, at 6.9 bits. And this has actually been seen in wet lab experiments along with exon skipping. The ASSEDA draws a structure, the ones that are perspective isoforms. Here's the natural site. Here's what happens when it gets abolished. It's predicting exon skipping. This is the splice form that extends by 87 nucleotides. And another one that's 133 nucleotides out, I believe. It wasn't seen by experimental conditions, but as you can see here, it is much weaker. This is the 3D plots showing the relative abundance. 17 is the natural, 1 is the highest ranked. So if you come to the 3D plot, number 17 before mutation is the most abundant, especially compared to number 1 and number 4, which were the other two potential splice forms. But after mutation, suddenly isoform 1 is much more abundant compared to isoform 17. And that's how you visualize this. One extra new option is to be able to visualize these splice forms using the UCSC genome browser. Now if we scroll down, we see where the BRCA1, see this has been skipped in one of the mRNAs, and we see all the different potential splice forms. Here's the natural site, here's the first ranked ones, here are the two that second and third ones that do not conform to natural splicing. You can see that it's shifted over. And here is the isoform 4, which is the 133 nucleotide downstream cryptic site. So I hope you can use this resource to better understand your experimental results. Thank you, and have a good day.